During this lecture, we'll be talking about the Hardy-Weinberg Law. Well, first of all, let's understand what the Hardy-Weinberg Law allows us to calculate, and then we'll dive into a small example. Well, first of all, the alleles and the genotype frequencies within a population, that's what the law says, is going to be remaining constant after a generation or many generations if certain conditions are respected. Now, there are many conditions that have to be respected, and we're going to be diving into them one by one. The conditions are as follows. First of all, the population follows the laws of Mendelian genetics. Second, the size of the population is infinity or goes towards infinity. Obviously, when we're talking about animals or even plants, it's quite impossible to reach this figure, or to reach the infinity level. So we're going to be just using the size of the population as large is actually good enough. Now, why is this important? Why is the fact that the size of the population must be large? Essentially, because the variation in the relative frequency of different genotypes in a small population is going to be increasing the chance of the disappearance of a couple of genes. Now, if these are going to be changing, there is going to be a genetic drift. The genes that are present, as we know, are going to be changed, relatively speaking, to the population. So if the population size was actually large, even these small changes that might occur will not make a massive difference. However, if the population was small, it is going to be making a difference. Now, no gene mutations, obviously, because just like we mentioned before, a mutation is going to be causing is going to be changing the alleles, and this is, we're going to be diving into that actually shortly in the next video. But briefly speaking, gene mutations can alter the Hardy-Weinberg Law. No natural selection, no migration into or out of the population. This is also to maintain the gene pool, because if we have a particular gene pool and an organism or even a member of the same species actually migrates into the population at hand, it is going to be altered, since there are different differences actually between the organisms that are present within a specific area at a specific time and a member of the same species that is actually staying at a different area. So it is going to be altering the gene pool in this manner. Also, making sure the mating is totally random, and this is going to be further discussed in a later video. What we are seeing here is a graph, and this graph actually allows us to calculate the frequencies of the homozygous dominant or recessive and the heterozygous. Now, this is the equation that we're going to be using for the most part when we're talking about the Hardy-Weinberg equilibrium or Hardy-Weinberg Law. And obviously, these are for the organisms or populations that are respecting the conditions mentioned before. And it is as follows. The P square, which is the frequency of the homozygous dominant, since P refers to, for example, if we're talking about a particular allele, then we're talking about the AA, that means the big A, and then plus 2pq, which is the frequency of the heterozygous genotype. Essentially, this is for the genotypes that have a dominant and a recessive, which is, for example, 2a, small a. And we've got plus q square, which is small a, small a. The frequencies of the genotypes that have these alleles. Now, this question can be raised, which was, what is going to be happening in the next generation, and how does the Hardy-Weinberg Law actually help us? Essentially, we have to calculate using the formula mentioned before, and the closer the number to 1, the less deviated the new generation is from the previous one. So, generally speaking, if the population is actually respecting the conditions that we mentioned before, the number is going to be extremely close to 1, almost by fractions. So this is going to be allowing us to know that there is a level of respect for this population to the Hardy-Weinberg Law. That means there are no mutations, the levels or the population size is actually large, no natural selection, etc. Now, in this final slide, what we are seeing here is a table that actually allows us to solve most questions pertaining to the population genetics when we're talking about the Hardy-Weinberg Law. Essentially, the genotypes is to the left, the BB, B small b, and the small b, and the total underneath it. We've got the number of the number of the observed organisms in a population, and this is going to be filling up. I purposely did not fill it up, so you can pick up questions from the internet in order to actually solve using this. Next, we've got the Fn, which is the frequency of the genotypes in this generation and refers to the generation at hand. Fn+1, which is the frequency of the genotypes in the generation that is next. Also, by the way, we can add a couple of Fn+2, Fn+3, and this is going to be for the next and next generation. We can easily calculate it. Then we've got the last column, which is the number of the expected N+1. Now, the number of the expected, I mean the number of the expected organisms present in a population for this particular genotype. Now, it's easy to calculate it because all we have to do is multiply the Fn+1 multiplied by the total number of the observed organisms in a population. And since we've got the frequency times the population, we will get the number of the expected organisms. Now, when in doubt, we can always use the chi-square test.